Welcome everyone. We recognize the land we're standing on today is the traditional territories of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis peoples. We also acknowledge that Ontario is covered by Treaty 13, signed by the Mississaugas of the Credit, and the Williams Treaty, signed by the multiple Mississaugas and Chippewa bands. Ontario Genomics is delighted to sponsor today's webinar on new frontiers in food innovation. I want to tell you a little bit about Ontario Genomics and some of our recent and upcoming initiatives. We're a not-for-profit organization leading the application of genomics and engineering biology-based solutions to drive economic growth, improve quality of life, and global leadership for Ontario. We work across three key economic sectors: agriculture and food, human health, and industrial biotech, and foster collaborative opportunities to collectively achieve our vision of healthy lives, a healthy economy, and a healthy planet through genomics innovations. We're funded federally and also provincially through the Ministry of Colleges and Universities, and since our inception have brought $2.7 billion in funding, over 230 projects, 500 partnerships, and more than 20,000 jobs to the province. We're always looking towards the future and new opportunities for Ontario. And for the past several years, I've been working in the cellular agriculture space, the biomanufacturing of various food products and bioreactors. This includes proteins such as dairy and eggs, ingredients such as flavorings and pigments, honey, chocolate, meats and seafood, and many others. Our recent cellular agriculture focus report that you see here highlighted that this space could be worth up to $12.5 billion annually and create over 140,000 for the future for Canada, and articulates three interconnected, actionable opportunities to realize this enormous economic potential. We partner with the Canadian Food Innovation Network for Accelerate On, that you also see here, Canada's first regional cellular agriculture competition. And last week, we announced 4 cutting-edge cell-focused food and beverage projects that are using genomics or engineering biology, and some of whom are speaking here today. We'll also hold our 4th Canada Symbio Conference in Toronto from May 31st to June 2nd. This conference brings together Canadian international stakeholders, lots of great panels and keynotes. We'd encourage you to check it out. The website link is also here, and to attend. For today's webinar, I'm delighted to introduce you to Bronwyn Wilton, Principal at Wilton Consulting Group and LSO board member. Bronwyn will moderate the discussion today and introduce you to today's fantastic guest speakers. Thanks so much.
Wonderful. Thank you, Elaine. Thank you for the introduction, and thank you to Ontario Genomics for sponsoring this webinar this morning. Yes, as Elaine mentioned, I'm an LSO board member and very pleased to be here this morning to moderate this panel on a very interesting topic that is going to, I'm sure, generate lots of questions this morning. So on that note, I'd like to remind all the participants to use the Q&A function to put your questions in there, and we will get to them after each speaker, and then hopefully we'll have time for a bit of an open panel discussion at the end. So don't be shy, put your questions in there, and we've got people here who will be able to answer them. So I'll introduce our next speaker, who is Steve George. Steve is the CEO and co-founder of Terra Bio. And Steve has a question to start us off with. What if we could turn our existing supply chains into circular economies without breaking the bank? That's a great question. Terra Bio has created a way to do just this with their upcycling platform. This platform sources local undervalued spent grains, a byproduct of beer production, to create solutions for the plant-based protein and precision fermentation industries. Find out how this team of chemical engineers and entrepreneurs are creating functional and competitive plant protein while also creating a highly sustainable feedstock for the growing fermentation industry. With just one process that can be placed anywhere in the world, this team is improving the scalability, sustainability, and circularity of existing supply chains to make environmentally friendly industrial inputs more accessible. So with that, I look forward to your presentation, and the microphone's over to you, Steve.
Thank you very much for the introduction. As Bronwyn said, I'm Steve George, CEO of Terra Bio, one of the co-founders. And really first, I'd like to start by just saying why we need a company like ours to exist even. And it basically has to do with our current paradigm of how we produce and consume materials. You know, we have this take, make, dump mentality. So essentially we take resources, whether that's minerals or water or land resources, we take it, we make it into whatever materials we want, we use it, and then we dump it, and it becomes useless at that point. And this has some real world consequences. You know, there's the figure on the right is something that I came across in the past week. It's quite shocking. It's essentially the Earth's boundaries, the resource boundaries. Essentially, it shows where we are exceeding the Earth's carrying capacity. And out of 9 boundaries, we are clearly exceeding at least 6 of them. Things like land, biosphere integrity, climate change, fresh water, critical things. And in the 3 others, which we don't have, which we're not exceeding, one of them is just because we have no information. This is quite shocking and obviously highly unsustainable, and this has implications for us. For instance, one of the shocking headlines that I found, on my searches is that Earth has lost a third of the arable land in the past 40 years, and what's remaining, a lot of it is actually highly degraded. That is tremendous, and that affects us directly. For instance, last year, due to a combination of factors, including climate change, Canada's pea harvest was reduced by somewhere around 40 to 50%. And that means higher prices for us. So this is where we want to come in and really connect our supply chain back into a circular economy. So what Terra wants to do is to create locally sourced and reliable ingredients. So reliable means we essentially have ingredients of the same quality as traditional ingredients, but except that these ingredients would be made through upcycling what we already have. So our vision for the future, for future process is, what is needed to get away from this take, make, dump mentality is any future production process needs to be circular. Essentially, it takes something that's already produced and it makes it into something useful. It could be a byproduct, whatever it is, make it into something useful. These processes need to be sustainable. It needs to impact the environment as little as possible. And it needs to be scalable now. We don't have time to implement exotic solutions. We need to be able to scale whatever we want right now. And scalability, of course, starts with having a lot of material to work with. And this is where Terra started looking at spent grains. So for instance, in terms of brewer spent grain, which is the byproduct of the beer brewing industry, mainly barley grains, there's about 39 million tons of this stuff available worldwide. And if we look at corn spent grains, which is essentially a lot of it comes from the distilling industry, whether for biofuels or spirits, whatever it is, there's about 36 million tons of this stuff. So quite a lot. And what we're doing is we're taking our process, our core platform process, implementing it on these different spent grains and trying to make various environmentally friendly products out of it. So we've started with brewers spent grain, and essentially we're producing, at our current state, we're producing two products out of it. The first one, and that's at the bottom. The first one is a high protein, a high content protein called Proteina that's made from brewer spent grains that we're marketing as an ingredient that we sell to other businesses for various applications. And also we produce a rich sugar mix, which we're positioning as fermentation feedstock, particularly for the emerging precision fermentation industry. This slide is just to reiterate what I just said, and to say that we really want to produce these using a model where our products would not be significantly more expensive than what is already available on the market. So we were looking at drop-in solutions, which people can take without a significant economic penalty. And this slide is just to say that we don't want to stop just at where we are. Our vision is a little more grandiose than that. We believe that there are many, there's many products that can come from each byproduct source, whether it's brewer spent grain, corn spent grains, whatever it is. And once we exploit the potential of, let's say, brewer spent grain, we can go to other feedstocks like corn spent grains, like okara, whatever it is, and we can valorize that as well. And there's a huge potential for this worldwide. Yes. So like I said, we have to start somewhere. So we started with brewer spent grains. We're splitting it into 2 products. The first one is Proteina, which, like I said, is our spent grain protein ingredient. We started by positioning this towards meat replacement applications, but now we're looking at other applications like specialty pet foods and also snack foods, things like that. And the way we're positioning this is that it meets this holy trinity of having a mild taste. It provides good texture to the foods that it's added to, and it's available at a price that's not significantly more than traditional ingredients like pea, soy, things like that. And it's all tied together with this story of sustainability, and that's at our very core. We've been doing a lot of work trying to establish, for this proteins, we've been trying to establish both market validation, which is essentially we try to establish product market fit. We're trying to improve the functionality of proteins, and we do that through various contract research organizations. So, for instance, through the Saskatchewan Food Centre and the University of Guelph, we are doing exactly that. We have experts working on improving the functionality of the protein for particular applications like meat replacement applications. We're thoroughly characterizing our protein, and we're also, developing prototypes for various applications so that we can support our marketing. One of the fun projects that we're actually doing is through the X Prize Feed the Next Billion competition, where we're working with an algae protein producer in Halifax called Small Food, and we're combining our two proteins together to create a fish fillet alternative. In particular, it's a herring fillet alternative that is meant to mimic the real thing in terms of taste, texture, and nutritional value, while being extremely, extremely sustainable. And we're very excited about this. The semifinals are actually in December of this year. The other thing that we're doing is essentially sending out our protein to customers, and they will try it out in their labs, in their applications. And we're waiting to hear back from 3 of them. Just to quantify a little bit of what the benefits of using this upcycling approach is, we are able to lower emissions essentially at every step of the chain. So when we take spent grain, brewer's spent grain, and we convert it into something useful, we're essentially lowering emissions. We're removing the emissions that come from, landfilling the spent grain or using it for other purposes. And then we convert it into a protein. And that protein, when we use it for food applications or whatever it is, it displaces traditional proteins, which lead to additional emission savings as well. So it's a win-win situation in every case. And the interesting thing is on the left, you have, it says shorter supply chains at economies of scale. And this is essentially a point that these spent grains are generated world over in massive quantities. So we believe that when we fully flesh out our vision, we will be able to essentially have much more local supply chains to use spent grains that are found locally and supply industries that are found locally. And that's our vision. In terms of our sugars, I mentioned that we produce sugars as a fermentation feedstock right now. It's essentially a rich mix of C5 and C6 sugars, which we actually initially produced as a byproduct of our process. And what we realized very quickly is that companies are hungry for new feedstock sources that are highly sustainable. This is a realization that we came to relatively recently. And so what we did is we actually took it and tried it out as a fermentation feedstock internally, where we were successful in growing microalgae on these sugar feedstocks. And then we got a little bit bolder. We sent it out to several companies, and they used their own industrial strains of mycelium and microalgae and they grew it on our sugars. And what they found is so far their tests show that we're as good or better than conventional feedstocks for these organisms. So we're very excited about the potential of this product. So just to introduce the team in very high level detail, myself and my co-founder, Ricardo, we come from very similar backgrounds. We're both chemical engineers as well as biologists with a lot of industry experience. And really we united in the same goal. We want to help circularize tomorrow's economy for as many products as we can. That's what we really want to do. And we're supported by a team of young, highly motivated people who share our vision. And that's a fantastic situation to be in. It really does take a village to support and grow an organization like ours. all of these organizations have contributed in some way to us, whether it's simply advice, a conversation, or funding. All these organizations have been a huge help to us. We do most of our work in the two organizations on the right over there, the Saskatchewan Food Industry Development Center and Lambton College. And at the bottom, we have two of our industry partners who we're allowed to say something about so far. So Wellington Brewery is, of course, a well-known brewery in Ontario, and Sophie's Bionutrients is a microalgae producer. And we're working on getting more industry partnerships. So the last page is essentially just saying, get in touch with us. We'd love to hear from you. We welcome new perspectives, and we always welcome if you have any advice for us. So that's all from me. Thank you.
Wonderful. Thank you so much, Steve. That was a really interesting presentation. And I know we have already one question in the Q&A for you. And please don't be shy, members of the audience. Type your questions in and we'll try and get to as many as possible. So our first question for you, Steve, is what testing is performed on spent grains prior to use as a raw material? Are they tested for mycotoxins, for example? And are there regulations in place for minimum levels of potentially harmful contaminants in these raw materials?
Mm-hmm. That is something that we're actively exploring right now. Our quality plan essentially, it focuses mainly on the collection of the spent grains. And that is exactly the question that we're defining right now. How do we maintain the quality of the spent grains all the way from when it comes out of a brewery immediately after the separation process that gets the spent grains, and how do we maintain that quality out to when we get it to our production facility? I don't have a specific answer for you, but toxins and bioburden is exactly the kind of thing we're looking at. And the other thing is, inherent in our process is essentially control steps that control this, the toxin content or bioburden content as well.
Oh, very good. Thank you. Yeah, it seems like an important part to be exploring and researching as you go.
Yes.
Thanks for that answer. Another question here is, do you have any projects utilizing edible gourmet or medicinal mushroom mycelium to use spent grains for the production of high quality complete protein or nutraceuticals?
So we don't. Partners do. It is simply for protein. Our partners simply use the mycelium for protein. That's not something we do. We merely produce the ingredients. We're one step behind.
Yep. Oh, great answer. So yeah, it takes the whole kind of value chain and to build up this as well in terms of products and final things. So great. Yes. Thanks for that, Steve. And I think in the interest of time, we'll turn to our last speaker. So hopefully we can have a few minutes left at the end for questions for everybody, but really interesting presentation. Thanks for sharing that today, Steve.
Thank you.
And our last, but not least panelist this morning is Dr. Valentin Fuga, co-founder of Cell Ag Tech Incorporated. Dr. Fulga is a serial entrepreneur and executive in the life sciences industry. Valentin is an MD who has established and led several companies from inception to major value inflection, including taking public one of the companies that he founded. During his career, he has climbed all the steps of a biotech career, starting as a research scientist and up to president and CEO level. As senior VP of development at one of the first cell therapy companies in the world, and then as CEO of a stem cell company, he has been intimately involved in the development and manufacturing of cell therapy products while working hand in hand with the FDA and Health Canada. Valentin was recognized as a technology pioneer by the World Economic Forum, and he is the inventor of more than 50 patents, the author of 15 scientific and medical articles, and has been an invited speaker at many prestigious scientific and business meetings. So we're very fortunate to have Valentin here with us this morning and to talk about Cell Ag Tech. So over to you.
Thank you very much for inviting me here to speak about Cell Ag Tech. Thank you to Life Science Ontario. Thank you to Ontario Genomics for sponsoring this session, and also for awarding us the Accelerate Ontario grant. I'm blushing now after saying also so many good things about me. I hope that I'll be able to present Cell Ag Tech and explain why we're doing and what we're doing, and I really look forward to giving you more information about us. So who are we? We are a seafood company. We're making seafood from stem cells like any other cellular agriculture company. And we're really aiming to becoming the global leader in production of sustainable cell-based seafood. We were established in 2020, just in the middle of or at the start of the pandemic. We have a small lab in Toronto, and we're focused on lean white fish. One of the reasons that we're working with lean white fish is because we're really trying to develop the myoblasts into the muscles, and not work with the adipose cells. We raised until now about $1.25 million, some of it being non-dilutive funding. We have already filed a PCT application in December 2021. We were selected as one of the semi-finalists of the X Prize almost a year ago. And we are focused on a hybrid product between cell-based and plant-based prototype, which you'll be able to see here. We're working in collaboration with the World Food Innovation Center on this product, and you'll hear in a moment that we're going to do a little bit more about that in the future. Why, in general, make seafood and not make meat? It was very interesting because when I convinced my co-founder Josh to work on this project, it took me about 2 months to convince him that it makes more sense to make fish rather than meat. He wanted to make sausage. He didn't like fish at that time. Now I think that he likes fish, and definitely he understands the reason why it makes more sense to make seafood rather than meat. Not because it's impossible, but because there are some very good commercial and also scientific reasons to make it. There are advantages to the industry. The production is not limited by location, and the costs of seafood in general it's quite high. There are no quotas in this case, in our case, of fishing seasons, and many other reasons for that. In addition, there's a very important issue with seafood. They are full of microplastics, hormones, and other contaminants. When you make seafood in the lab, this is not going to be a problem. There's no animal cruelty, of course, like any other cellular agriculture project. And there are more advantages for both the industry and the consumers. It's sustainable. Everybody knows that this is going to be much more sustainable. We don't think that we're going to take the place of everything else, of all the other agriculture fields, but we will be an addition, and an important addition to sustainability of production of food. Our production will be only making the desired cuts. We will make fish fillets without the scales, without the fins, without anything else that is not edible in a fish. And we think that they will be also much longer lasting because first of all, they will be sterile, and they will be probably in a much better condition instead of what's available today. And many of the production, a lot of the production of especially seafood has a big issue with spoilage. This is a very simple slide that tries to explain that seafood market is a big market in the world. That's not, we don't need to speak about it anymore. It's clear that this is an important market, not less than anything else. And it is limited by the availability of certain fish species. I've been requested to speak about the challenges and the opportunities in this field. So I'm trying to comply with the request. And the first thing that I would like to speak about is the scientific and the technological challenges. And for people that are involved in this field, I think that people know that there are 4 major issues that have to be solved in order to have a viable and a sustainable cell agricultural product. One is the need for a cell line, and to have an immortalized, a continuous cell line that continues to produce cells in a sustainable and also in a very consistent way. That's something that, as you can see here, we already have, and I'll share with you a little bit more information later. In addition, we need to eliminate animal-derived ingredients from culture medium. You cannot use fetal bovine serum, certainly, in a cellular agriculture product, because again, it doesn't make sense to use something that will not use animal-derived products and use animal-derived reagents in growing the cells. Also, to me at least, it doesn't make sense that fish will use cow derivatives in order to grow. So that's another important issue that we have to deal with, and we are in the process of working on this and eliminating animal-derived ingredients from the culture medium. Another important challenge is manufacturing and scaling up of the manufacturing process. That's something that we started lately in collaboration with CCRM, Center for Commercialization of Regenerative Medicine. And we think that this is one of the most important things, because in order to get to a decent price for the product that we develop, we will need to have a scale up of scaling up the manufacturing process in a way that it's much cheaper than it is done today. And last but not least, a challenge that we haven't tackled yet is developing a structured 3D culture of the product. It can be either a steak or a fish fillet, as in our case. Where are we standing now? And that's something that I wanted to share with you until now. First of all, as I mentioned before, our technology is patent pending, and we have a quite extensive patent that is going to become public domain very soon. So I'm not that concerned about disclosing things that are proprietary, because we are protected, and we think that also because our team is quite experienced in developing processes for the pharmaceutical, for the biotechnology industry. We know how important patenting is in fields related to science. We already had the pre-submission meeting with Health Canada, which was very successful. They were very supportive, and I think that they will continue to be very supportive once we move forward on our development process. We, as I mentioned before, we established already 2 myogenic continuous lines, and we still have another 3 additional lines that have the potential to become immortalized. They have been passaged over 100 times, and they are, just as I always call them, mini robots that continue to proliferate at the time that we need them and at the level that we need them. This was done with no genetic modification, because we're very aware of the fact that some of the regulatory authorities are not going to allow us to use genetic modification in order to use in our products. And this is how our mini robots look under the microscope. And in addition to that, we have optimized the condition in which we expand our cells. And just to give an example, some of the conditions were related to the seeding density. And as you can see here, there's a very strong relationship, opposite relationship, between the doubling time and the seeding density. The lower the seeding density, the lower the seeding density, the lower the doubling time. And this is one of the things that we were able to optimize while we developed our process, and we continue to use different optimization processes in order to make our process as efficient as possible. Again, where we're standing now, we have a cell bank with over 300 cryopreserved samples that retain their high viability and proliferation differentiation rate. We are differentiating our cells also into adult myotubes, so we can definitely know if the cells that we're growing have the potential of really turning into meat. This is one of our first myotubes that we differentiated, and we were able to show that they also exhibit markers that are specific for myotubes. So we know that they don't only look like myotubes, they also have the markers specific for this. And these myotubes are definitely much higher in protein and also heavier. So the product can become more viable and more sustainable in terms of efficiency when we differentiate ourselves. What are our ongoing programs? As I mentioned before, we are developing a non-animal derived ingredient medium. We're working in eliminating FBS completely. At least at this stage, we were able to go to lower than a third from what we started with the FBS initially. And I'm just going to show you, unfortunately, I won't be able to tell you exactly what the supplement is, but I can show you that, as you can see here, this is the normal, this is the control, this is the normal concentration of FBS that we're using. In all these cultures that we have here, the concentration of the FBS was lowered like 10%. And as you can see here, the supplement increases the yield of the expansion rate significantly. It is significantly statistically significant, sorry. Statistically significant when we use higher concentrations and almost at the same level as the regular control. So we're very excited about the introduction of this supplement, and we think that this is going to be a real breakthrough, not only in the seafood market, but in the seafood cellular agriculture market, but also for other potential uses in the cellular agriculture. We're very excited about this, and we're moving forward in improving this even further. In addition to that, we are now working, as I mentioned before, with CCRM in selecting the cells that can grow in 3D cultures in bioreactors and scaling up our manufacturing process. And we're working with the World Food Innovation Center in developing a more advanced food prototype. Our plans for the future, obviously to develop additional cell lines, to develop fish growth factors, specific fish growth factors that practically don't exist at this stage, that we think that are going to be more efficient and also cheaper to develop. We're in discussions with a few companies that with which we can collaborate in order to develop these fish growth factors. Increase obviously the scale-up manufacture to hundreds of liters once we overcome the proof of concept that we're working with CCRM, and optimize the cell muscle, the muscle cell differentiation process, which is something that we're working, we will be working in the future. And this is based on a raise of $3 million that we're working on these days. So anybody that wants to invest, we're more than happy to receive. Going back to the slide that I showed you before, the challenges, now there are quite a few challenges from the commercial and business perspectives, and the first one is funding. This is an emerging industry, a very novel industry, and even though there are not many, or maybe because there are not many cellular agriculture companies in Canada, it's not easy to get funding. We're very happy that we were able to receive the grant from Ontario Genomics and Canadian Food Innovation Network, because this will help us to continue our work. And that's why we're really trying to get more funding, and spreading the word about cellular agriculture is also important from this perspective. Challenges, lab space. It's quite difficult to find the lab space, especially for a cellular agriculture company, which is not therapeutics. It's something in between. So people have difficulty in understanding what we're doing. So again, spreading the word is important. And the public recognition of the potential. People don't know about this. And this is also an issue that can resonate with the food that we're going to develop in the future. The regulatory framework, it's still in development. We know that Health Canada is going to regulate our product as a novel foods, but still there are quite a few questions in how cellular agriculture products are going to be regulated. There are opportunities. First of all, there's cell therapy, there's regenerative medicine, and a lot have been developed there. So we think that there are lots of concepts that can be taken from there. And of course, the ability to develop new products and develop these products not only in terms of for the use of people, but also developing factors for cellular agriculture. That's an opportunity that could increase this industry, and it's already taken by some of the companies like Future Fields that were already mentioned in the past. And I think that Canada, as a leader in agriculture, should become also a leader in cellular agriculture as well. There are the resources, and it's something that we should work on in the future. Last but definitely not least, we are a small team, and I have to thank everybody in this team, and also to the people in our advisory board for their help. Without their dedication and without their hard work, we wouldn't be able to be in the situation that we are today after 2 years. They are people that have experience with cells, have experience with myoblasts, have experience with fish cells, have experience in bioinformatics, and of course our advisory board that can help us from many other aspects of this novel industry. And we're very, very grateful for all their help. So a big thank you to all of them. And now a big thank you to everybody that was here until now. Thank you.
Wonderful. Thank you so much. That was a really interesting presentation again. Didn't disappoint at all. I'm very interested in the again, the consumer acceptance of the fish from a cell lab. So I'm curious about that. And I'm also curious about how long this might be till I know you mentioned you're in pre-consultations with Health Canada and things like that, but when could I possibly walk into a grocery store and buy this for dinner with my friends and family?
You know, that's a difficult question, and that I don't want to overpromise and under-deliver, okay? So let's not put numbers or timelines into this, but what we hope is that we'll be able to be limited commercial in about a year and a half, 2 years from now, if we raise the next round of financing. Limited commercial being able to sell to high-end restaurants or delis or places like this. Definitely not, you won't be able to find our stuff in any grocery store, but we think that it will be available. I think that's something that we will be able to do it. We're very efficient in our research process, in our developing process. So I think that we will be able to reach the stage in maximum 24 months.
That's great. I look forward to that. I have a really great question here that I might open up to all three of the speakers, and it might be our only question we can get to from the audience, but currently without dedicated courses or training opportunities, what is the path into this industry for interested students? Is there an awareness among highly qualified personnel that careers in cell ag are an opportunity? So I think that's a really great question. So Dr. Fulga, I'll start with you, and then maybe I'll turn to Frank for comments on that.
You know, we were able to get people into our company quite easily. They were interested, and and one of the questions that I asked everybody when I interviewed them was, you know, you're a scientist, you're working with cells, don't you want to be in the therapeutic field? It's much more heroic than making food. And they thought that it's as interesting and as exciting as making therapeutics. So this is, I think, more people know about it, they will come into this industry, and there's a lot of room for this industry. So I think that's something that they will definitely take as an option. Are they aware of this? I think that more and more people are aware of that, and they will continue to learn about it because there's much more noise made about it than it was, I don't know, 2 years ago when we started. So there are more companies here in Toronto, as I'm very happy to hear. So hopefully there will be people coming into this industry.
Great, thank you. Frank, do you want to comment on that piece?
Yeah, for sure. I agree with Valentin's sentiments, and there are actually more and more courses around the world for cellular agriculture. Actually, I think Singapore, the National University of Singapore, released a graduate course for advanced meat alternatives. So if anyone interested, they can take a graduate course there. And I think Tufts University, with their recent funding from the U.S. government, they also have a few more graduate courses for alternative meats. And also wanted to say that, you know, like most people who went into this industry didn't study cellular agriculture. And interestingly, too, if you look at the co-founders of a lot of the major mammalian cultured meats, and also wild type in the U.S., their co-founders are actually all cardiologists. what that means, I don't know, but there's definitely a lot of translatability from life sciences into this industry, and it's not just limited to life sciences, right? We need really good engineers, we need really good bioengineers, machine learning scientists. Anything cutting edge we can apply to the way that we produce our meats. And you know, the more people we have, the more brain power we have, right? So we're always looking forward to growing the industry and growing our teams.
Awesome, that's excellent. Thanks for that, those insights. And I'm looking at our time, and we are at the end, near the end of our hour. So I'm going to turn things over to Jason Field from Life Sciences Ontario for some closing comments.
Thank you, Bronwyn. Fantastic job moderating this panel. I really appreciate you taking the lead on this. And thank you to all of our speakers. A fantastic session. I think this has been really eye-opening for many of us. We hear about these new technologies and we see them coming to market slowly, and to get this inside view at the challenges that you face and the technologies that are utilized has been absolutely fabulous. So thank you all. A few quick announcements, and if you could pull up the slides, Brian, great. So our next event is on May 27th. That's our member update. If you are a member of LSO and interested in what's happening within our world, we'll be giving an update to all of our members at that time. The next in this series of our breakfast sessions is on May 31st. And Dr. Fulga talked about this, the challenges of finding lab space. And so we'll be talking about that, and we'll have some real life case studies and examples from Jeff Evans at Eurofins and at Alfora Eurofins in Mississauga, as well as Scott Mulligan at Ellington Corporate Real Estate Advisors. And then we'll continue our member marathon on June 23rd. Our scholarship program is open. So if you know undergraduates, college students that are interested in applying, please direct them to our website. And finally, a thank you to our platinum sponsors, which are shown here, and our gold sponsors, and of course our silver sponsors. And a special thank you to today's event sponsor, Ontario Genomics. We appreciate the support, and thank you all for attending, and we look forward to seeing you soon. Stay safe. Have a great day, everyone.